Good morning, my dear students. I'm going to deliver my course on cell signaling and molecular biology and cell signaling. Today, I'm going to my first lecture, and that is in molecular molecules and life, part one. I'm Mehdi Hayas Shahi, senior assistant professor at Interdisciplinary Brain Research Center, Faculty of Medicine, Aligarh Muslim University, Aligarh. This topic is very important, and because we wanted to know how molecules interact and also how it is important for the life. That's why we wanted to explain in this lecture. Yeah, the content of this lecture is biomolecules. There's a lot of biomolecules which I am focused in this lecture, especially the DNA, deoxyribonucleic acid, and then biological diversity and the complexity, how biological diversity and complexity causes. Then we focus on evolution of organism, how organisms are evolved, means how they organized. Then we discuss about chemistry of the biomolecules, means how biomolecules act in a solution, how the chemistry looks. Then we have discussed about DNA double strand, the characteristics of the double strand, how it acts, how the influence with the water and different pH and solution. Then we discuss different types of chemical bond. The references of the two references in this lecture is first is the biochemistry well-known book from Lubert Strad, 8th editions, and the second references is a journey from amino acids to protein by Saad Tayyib and Amru Nasrullah Boyce. Molecules and life. This is very important. And the life which we have here is also depend on molecules, what molecules we have. And that's why life is based on different types of biological molecules. The different type of biological molecules, they builds the block of life. And all living organisms function at a molecular, the living organisms, they're all molecular level functions, and then we study the molecules and all sort of interactions, and it's covered in biochemistry, because biochemistry is the part, the combination of chemistry and the biology. So mainly the biomolecules which we discuss in this lecture is the DNA, but also the other in lectures we discuss about the proteins, lipids and carbohydrates, and also we discuss the cellular process. What are the cellular process? Is the metabolism and cell signaling. These are the very important cellular process we wanted to discuss in this lecture. Why a study of molecules is important? Why it is very important? Because it's provide a understanding of life at molecular levels. Because whatever we see, any organism or any plants or any systems, it has molecules which govern and control the life process. And also it is essential to know about different diseases. If there is any disorganizations or disruption in the molecules, then they has causing disease, and then we have to know how to diagnose the disease. We wanted to understand molecular level, molecular mechanisms, interactions of the molecules, and then based on those interactions, we understand the functionality of the life. Then we design a medicine, and then with this medicine, we know that then we can have to know some novel medicine to drug discovery, and also we know the developmental process of the life. That's why understanding of molecule is very important to understand the life and the process of that. And also there's an application that's in agriculture. There's very vast applications, because it improves the crop productions, increase the crop productions, also develop many resistant crop varieties that gives a lot of yields even in the adverse conditions. Then also we understand environmental sciences, because environment is very important, the ecosystem, and the molecular level also influencing, then there's a co-ecosystem existing with different organisms that we wanted to understand. Then also the forensics in the also important of the molecules in the DNA fingerprintings, and also with the help of that we are unable to understand many events, that whatever crime scenes, all sort of things we can study at molecular level with the DNA fingerprintings. Then also this molecule interactions also important in biotechnology and also in genetic engineering, because biotechnology we can use the biological tools and technology for the applications in the biological system, for the welfare of the human. And also we do sort of genetic engineering to modify the plasmids, modify the bacterial plasmids, and all modification. We also modified the viral particles for a useful therapeutic approach. That's very important. Then biological diversity and the similarity, what it means. Whatever we see in the biological system, we see different range of organisms, all different diverse organisms. But if we see them at molecular level, then we find a kind of similarity in that molecular level in different organisms. The best examples we can take Tata box binding protein. We know this is very important protein for the transcription process and bind to many transcription factors in these regions. So see, this is three different organisms we have. We have Sulfolobus archaea. This is a very old bacteria. Then we have this Arabidopsis thaliana, is a very old plant, and then Homo sapiens. We are so all three phenotypically is different. These three distinct organisms have phenotypically different, but if we see their Tata box binding protein, almost all proteins are, if you see, all are same. All are same. If you see this, all are similarity in that. Why it is similarity? Because the function of molecular level are similar. We have phenotypically different, looks like that's very old and new or whatever evolved, but the functionality of the molecules at molecular level, at cellular levels are very different, very similar, and that's why the molecules like Tata box binding proteins or many other proteins are very similar. This is the reasons we can say we have biological diversity and also we have similarity at molecular level. Now, a possible timeline for biochemical evolutions. Evolution is a very fundamental process and it is a very slow process and it takes a lot of time, like millions of billions of years and all that. So what exactly? Things had evolved, organism has evolved. Then we have a life exists in various forms and size, different forms and size, like microorganisms. We cannot see with naked eye mostly the microorganisms. We need to help with a microscope. The microorganisms like bacteria and yeast, we need to see. And then we have a complex organism like plants, animals and humans. These are complex organisms we can see with naked eye and all that. They survive in a very diverse environment and is including very extreme, very hot temperature and also very cold, low temperature. So they survive in a very diverse environment and because of that reason they are surviving. So they have some characteristics on that. They develop some characteristics at molecular level that support their survival. We know that Earth evolved almost 4.5 billion years ago and then formed, and then we have microorganisms developing 3.5 billion. Then we have billions of years takes like cells with nuclei between 2.5 to 2 billion years ago. Then we oxygen forming atmosphere 0.2 to 1.5 billion years ago. Then we have macroscopic organisms we can see like 0.5 billion organisms. Then we dinosaur and also we have human arise very early. So roughly we can say that 3.5 billion years ago Earth begins, like life begins on the Earth. This is the evolutionary path. The biological evolution. What is biological evolutions? We have already explained diverse kind of organisms from bacteria to the elephant human beings. They all diverse and they survive in a different environment. But it's very interesting to know that their metabolic pathway is very similar if we compare it with the bacteria. For example, E. coli, Escherichia coli and human beings, how it looks. The glucose metabolism with the help of oxygen, and what they produce carbon dioxide and water, and they originated from the common ancestor. That's a biological evolutions. They have the same metabolic pathway for the glucose metabolism and they produce the same product in all human and bacteria. The tree of life is very important on the basis of biochemical characteristics, because that's very important at a molecular level. All organisms are divided into three fundamental groups. First, we can say eukaryotes. They are multicellular well-defined nucleus. Nucleus is well-defined nucleus. Then prokaryotes, they lacks a nucleus, means there's no nucleus in this prokaryotes. There's no nucleus, and then this prokaryote further divided in two groups by Waals in 1970 say that's called bacteria and archaea. So we have here the common origin here. Then we have three groups: bacteria, then we have archaea, and then we have eukaryotes. But both bacteria and archaea, they diversified in a very early stage, and then further from archaea eukaryote forms, and they have well-defined multicellular and well-defined nucleus in the case of eukarya, but not in bacteria and archaea. And we consider approx this evolution, this evolutionary path in common ancestor is approximately 3.5 billion years ago. 3.5 billion years ago, and they form the bottom of this tree in the modern world of the top. Biological diversity and the complexity. This is very important, because whatever the diversity we have, whatever the evolution happened, their body become complex, molecules become complex, then all living organisms share common features. What are the common features they share? Built from the cells, all organisms, either single cellular like prokaryotes and the eukaryotes, multicellular, they are built from cells. Second is very important part. They have genetic materials, that is DNA, and use as a genetic materials except some exceptional in viruses. And they all have very similar 20 amino acids which form the proteins. 20 amino acids form the proteins. Then also they have same or similar metabolic pathway. Like example already explained, the glucose metabolism. So this is the characteristics having biological diversity and also their complexity and the changes. Biomolecules. The molecules are really divided, means two major class of molecules we discuss. The macromolecules, that is protein and nucleic acids. Then metabolites, that is glucose and glycerol. And very interesting that a structure and the function of these molecules are highly conserved. Means that evolution happened in the organisms, they have complexities, but the structure and the function is almost conserved in the most of the organism. Now biomolecules, the chemical molecules is carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. These are chemical molecules, and they further form the molecules in a small molecules, then the large molecules. What the composition is small molecules is amino acid, which is responsible for the proteins and the enzyme when they mold, the protein and enzymes. Then we have nucleotides. These are responsible for the genetic materials. Then we have sugars. This is responsible for energy source and extracellular structure. Whatever the structure we have extracellular. Then we have fatty acids, which supports and storage. Then we have vitamins, its role in enzymatic functions. So these are the small molecules and they form function. Then finally these molecules, protein enzymes, form the polypeptides called proteins. Genetic materials is the nucleic acids, and energy source extra molecule is a carbohydrate, and this is a fat and fats and the vitamins. So these are the molecules features. Now the DNA is very important molecules because it's genetic materials and very conserved, very stable molecules. And now we have discussed about DNA, why this is important molecules. Illustrate the interplay between form and functions. DNA is a universal genetic materials in all living organisms. We well known that DNA is universal genetic materials in all living organisms. And it stores the genetic informations, means it's transformed from the parents and then from parents to the offspring and then offspring to further offspring. So they store DNA. The first discovery in 1940, the DNA identified as a genetic material, and then finally in 1953, after 13 years, the structure of the DNA is proposed, and that was by James Watson and Francis Crick in 1953, they demonstrated that DNA is a double helix structure. And why it is as a fundamental molecules? Because DNA is very stable and efficient to store the information. DNA structure enables efficient and stable information. And DNA is a linear polymer made of repeated uniform nucleotides. That's nucleotides is like adenine, cytosine, guanine, and thymine. These are 4 nucleotides. DNA has also the direction. That direction is 5 to 3 primes. And each nucleotides contain deoxyribose, then we have deoxyribose, then we have a phosphate group, and then nitrogen bases. And these are the bases: adenine, cytosine, guanine, and thymine. These are the 4 bases in the DNA. And also DNA consists, already mentioned, consists of 2 anti-parallel strands. That is an anti-parallel strand, and these form double helix. This form the helical structure, form the double helix. And this is the backbone. This is also called backbone of the helix. And they have in the backbone is made of sugar and phosphate. Sugar molecules and phosphate. This backbone form, and in this backbone there is a bases attached. Base 1, base 2, base 3. These bases are attached, so they're inside the backbone. And each unit of polymeric structure composed of sugar, deoxyribose, phosphate, and a variable nitrogen base. That is the structure of polymeric structure, and that's protrudes from the sugar phosphate backbone. So this bases protrude from the sugar and phosphorus backbone. The basis. And the base pairing is very important. Backbone is outside. This is outside the backbone, and then we have a bases inside there. Bases like adenine, thymine, cytosine, guanine. The bases inside the backbone. Base pairing rule, very specifically base pairing rule. Adenine is paired with thymine, that is double hydrogen bond, and guanine is paired with cytosine, that is triple 3 hydrogen bonds. And importance of hydrogen bond, why is it important in here, hydrogen bond? Because it is weak but specific and reversible. That's very important. It is weak but specific and reversible. And it provides stability and the flexibility. That is important because we need like DNA need flexibility and also stability. DNA stores genetic informations. The sequence of base is ATGC as a genetic code, and there's no restriction of the sequences. If there's no restriction, E comes after E, there's possibility of T also, C and GC. There's no restriction the sequences. Allow vast information storage. That is a characteristics that. And DNA also determine the RNA sequences, because when DNA transcribed, then this RNA formation, and also further RNA determine the protein structure and also sometimes functions, because then from RNA there is a translations and the protein structure definitions. And they have the complementary base pair already mentioned, A pair with T and G pair with C. That is also called Watson-Crick base pairs. Then is very unique characteristic. The sequence of the one strand determine the other. That's very important. It means that one strand is complement to the other strand. Suppose it is here T, so it is sure that other strand has thymine, that is T. A is adenine, the thymine. Then if other strand is guanine, G, is sure that it has cytosine. So one strand or one base determine the other base. It's very clear. Now the 2 strands of DNA combine to form double helix. A 2 strands of DNA combine to form double helix. And the bases are held for the hydrogen bond, already explained, which is very crucial for the biochemical system. That's why the hydrogen bond is there. And the three-dimensional structure of DNA is beautifully illustrate to the close connection between molecular form and function. This three-dimensional structure itself explaining that molecular form and function of the DNA. DNA structure explain heredity and the storage information. What is his heredity? Heredity means the transfer of a information, genetic information from one parents to the offspring. This is a transfer that's called hereditary informations, and it is stored in that DNA. And that's why the bases are very compatible for a storage of information, and they have same safe, and this is very important. When A pair be T and G pair be C, they are the same safe, and that's why they equally fit in the double helix. DNA sequence determine the RNA sequences and the protein molecules. That's very clear. And sequences of the basis of one strain determine the other strain, which I'll explain that. This is the one strain and this is the another strain. What happened? Nucleosynthesize. Suppose G is here, so very much sure C is here. If A is here, T is here, G is here, C is here. So you can see this is the complementary to each other. The DNA replication, this is a DNA replication process. Each strand act as a template. Means if each strand of the DNA act as a template for the other strand. When Watson and Crick identify the helical structure of the DNA, they very much things to mention that when they identify, they wrote, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic materials. When they reveal a specific pairing as a helical structure, and that specific pairing structure reveal that this materials has the characteristics of copying of our genetic material. That is a very unique structure, and that's why it's revealing itself. Now, concept from chemistry explain the properties of biological molecules. Now, this the types of the chemical bonds. Then we discuss the structure of water, because water is a universal solvent and it influences the structure of the molecules. Then we have first and second law of thermodynamics. That's very important. It's all system. We have to follow the thermodynamic rules, and system follow also thermodynamic rules. Then we'll discuss principles of acid and base chemistry. The chemical concept helped explain the biological process like the DNA helix formation. What the chemical process helping in the double helical formation, that is also influenced by the different molecular interactions. DNA double helix formation, a classical structure functional example. Complementary DNA strand can spontaneously form a double helix, means that it is very unique because the DNA one strand find the complementary strand, they form double helix, very unique characteristics. Suppose we have a sequence like here CGATTATT, CGATTAT. So then the complementary sequence of this is GCATATAT. So again, when the two single strand complement to each other, they mix, they form the double helix. This is very unique characteristics. So individually they exist in the single strand, but whenever they mix the complementary, they form double and stable double helix. And this reaction is almost near to complete. There's no hindrance. And the force, what are the force driving this interaction? That's a chemical bond, then the energetics, thermodynamics, and also the solution condition. What is the pH and ions involved in that condition? So DNA strand binds due to chemical interaction and it is energetically favorability. Means that whenever the binding between DNA single to double strand transition is a energetically favorable conditions. Now the chemical bonds, the two types of mainly chemical bonds. One is covalent bonds and another is non-covalent bonds, and they are important for a structure and stability of the biological molecules. Covalent bonds is the strongest bond, and it is formed by sharing of the pair of electron between the adjacent molecule. Its example is here. The single carbon binds covalent, and we have a double carbon covalent and the triple carbon covalent bonding. So single carbon is weaker than the double, and double is weaker than single carbon is weaker than double, and double is weaker than the triple. So the triple covalent bond is more stronger than the second and first. The bond property is the length is 1.5 angstrom, and then bond energy is 350 kilojoule per mole, and it is normally in the DNA, especially the adenine, they have in the resonance form, adenine, the new base in the resonance forms. Some molecules have multiple binding forms and is called example adenine, and it is not in single, not in double, is a hybrid mode in the form of resonance. You see, this is a double here, but this is single here, not double is here. So they is in resonance form. This is a characteristics of DNA molecule base adenine is a double structure. The true structure is a hybrid resonance form of single and double, and it increases the molecular stability, and that's important, or influences the bond length and the strength. That's why this is a single bond between two methyl groups, and its strength length is 1.34 angstrom, and this adenine absorbed bond length is 1.40 because this is between the single 1.34 and 1.54. That's why the length, bond length is here 1.4 angstrom here, because it is resonance structure. That's a 1.4 between this. This is very important for the stability and for the influence the resonance form. Chemical bond is a non-covalent bond. We discussed the covalent, now it comes to the non-covalent bonds. These bonds are weaker than covalent bond, but is very essential for to maintain the double helix of the formation of double helix. Then also important for the protein folding, and then also important for molecular interactions. They are weak individually but strong collectively. It's very characteristic. A single weak, they are weak, non-covalent bond, but multiple covalent bond, they are very strong collectively, and they provide flexibility and also specificity. This is the characteristic of non-covalent bond. And it's very important, they are also influenced by the water molecules as a solvent, and is essential. This is essential for biochemical reactions, all non-covalent bonds, interaction with water, and then also essential for a structure of the molecule. This maintain the structure of the molecule. Their dynamics of the structure of the molecules is important. Now, the four types of non-covalent bonds which we need to discuss in this: ionic interactions, ionic bond interactions, then we have a hydrogen bond, then we have a van der Waals interactions, and then we have a hydrophobic interactions. Now the next non-covalent interaction is a hydrogen bond. It is the attraction of the partial hydrogen bonded on oxygen or nitrogen, and a lone pair or another electronegative atom, Hydrogen bonds are responsible for a specific base pair formation in the DNA double helix, which already explained. A has a double bond with thymine T, and G is a guanine, has a triple bond with cytosine. So these are the hydrogen base pair, a specific bonding. A is always paired with T, not with G and C, and then same case for T always bind with the A, not with G and C. And the hydrogen bond, the hydrogen atom is partially shared between two electronegative atoms such as nitrogen and oxygen. So these are the two molecules, nitrogen and oxygen, they share the hydrogen bond. And the energy range is 4 to 20 kilojoules per mole, and it is longer than covalent bond, that is 1.5 to 2.6 angstrom. And hydrogen bond is very important because it is responsible for many of the property of water that makes the excellent solution, because we know that hydrogen molecule, water molecule is hydrogen bonded, and that's why it is the best solvent, and it's also an excellent solvent. So see, this is the hydrogen bond donor, it's H, and the green color is a hydrogen bond with nitrogen is acceptors, hydrogen bond acceptors. In this case again H is a donor and oxygen is acceptors. In this case H is the donor, hydrogen bond, and N is the acceptors, and then again H is the acceptor and O is the acceptor. A is the donor, H is the donor, and O is the acceptor. Hydrogen bonds are in green colors. This condition. Now another non-covalent bond that is also very important, and that is the van der Waals interactions. What is the van der Waals interactions? It is the weak interactions. If you compare with other interactions, it's the weak interactions arising from the transient dipoles between nearby atom. If there's a two atom nearby a certain distance in the range of van der Waals, they have interactions that's called van der Waals interactions. What happened? Van der Waals interactions occur when the distribution of electric charge around an atom influences. Means if there is a distribution of electric charges, electronic charges around the atom is distributed. At any distance, the charge distribution is not perfectly symmetrical. Not perfectly symmetrical. The distribution of charge is not perfectly symmetrical. The atoms and their neighbors, attract to one another, and this attraction increases as atom come closer. Means atom come closer in a particular distance, then they attract to each other until they are separated by van der Waals distance. Means that if they are in a range of particular distance, that's called van der Waals distance, they are attracted. If they are further closer than van der Waals distance, they are repulses. This example explained in the graph. This is the y-axis and this is x-axis. Y-axis we have from below zero the attractions, and then below zero repulsion, and this is energy, and they have a distance. So what happened? If a molecules comes closer to each other and in a distance, that's called this distance. This distance. It's called van der Waals contact distance. They have a maximum attractions in this range. But if they comes closer than this distance, they have a repulsion. See, this is a repulsion. From this is the distance, they have attraction, maximum attractions. But if they move closer to that van der Waals distance, they have repulsions. The same conditions, if they are farther, there is no attraction, but there is no repulsion too. No attraction, but no repulsions there. But they come closer, they repulsion. So they have in a particular distance, they are attracted, and that's called van der Waals contact distance. At a shorter than the van der Waals contact, very strong repulsive forces become dominant because of outer electrons clots the two atom overlap. Means outer electron overlapping, and that's why there is a repulsion because electron negative charges, and they have energy is 2.4 kilojoule per mole. Now, before going to the hydrophobic for last non-covalent interaction, we have to explain the property of water. That's very important. Water is a primary solvent of life. We know that our body is more than 70% of the water. The water is very important for the survival. And we know that water is polar molecule because it has two hydrogen and one oxygen. Oxygen is negatively charges and hydrogen are polar, and they have it tilted like this. So these are polar molecules. Oxygen has partial negative and hydrogen has partial positive. And they form electric dipole. This is the condition of dipole. One is negative and one is positive. That's called electric dipole. And the very interesting part also, water is highly cohesive molecules. Highly cohesive. Why? Because they are linked with hydrogen bonds. If you see, this is the water molecules, all are linked with hydrogen bonds. all greens you can see, their links very systematically, their links, that's a cohesive characteristics. It's this example of ice structure, ice, and also they have very high dielectric constant, that is almost 80, that is very high. And the importance, biological importance, it dissolves the polar and charged molecules because of hydrogen bonding, and then this stabilize the ions via ionic interactions, and also stabilize the ions these ionic interactions, and also enables of hydrogen bindings in the molecules, then this make very stability and also dissolve the molecules because they form hydrogen bonding with different molecules of polar and charged molecules. Now, the non-covalent bond is hydrophobic interaction. That's why we explained first the water characteristics. What is the hydrophobic interactions? It is the association of non-polar group in water to reduce the disruption of hydrogen bonded water structure. Means is the association of non-polar group, and that's cause is called hydrophobic. Means they avoid hydrophobic. Hydro is a water, phobic interaction. Hydrophilic means they are close to water, but in this case it is hydrophobic, means avoiding the water. And that's why the association of non-polar groups in water to reduce the destruction of hydrogen bonded structures. What a hydrophobic effect? This hydrophobic effect arises from the property of water. And what happens when the non-polar molecules, if suppose non-polar molecules, they do not form hydrogen bond and they do not interact favorably with water. So what happens if any non-polar molecules we add in a water solvent? What happens? Non-polar molecule bonded a cage around water molecule, the cage around non-polar molecules. This is a one molecules and this is the second molecules. Cage around means water molecules cage around, not because there's not finding any hydrogen bonding. That's why they cage around the non-polar molecules. So what happened? So what happened? Because of this cage around, the aggregation of non-polar molecules leads to release of water molecules. So what happened? So then what happened? Then this non-polar molecule tried to interact. So they wanted to break the water cages around. So these non-polar molecules interacted with each other, and that's due to the hydrophobic interactions. So what happened? This water molecule release, and then further they form the cage around these two non-polar molecules. Due to hydrophobic interaction, they bind it together and water molecules are released. So what happened? Initially interacting with non-sulfur in the bulk water. The aggregation of non-polar groups in water leads to release of the water molecules. Means they are releasing the water molecules and this reduces the entropy disorderness. When they form the cage, they reduce the entropies, but when they release the water molecules, they increase the entropy. This molecule interact together. Thank you so much.